And thank you all for joining. I'm excited to talk about this, and I think I'm feeling that there's a lot of excitement and enthusiasm of people out there for molecular pathology, so that's good. So as the title implies, this will be sort of on the basics of some of the molecular pathology techniques that you guys are probably seeing out there. And I just want to emphasize from the start that this will be mostly centered on really foundational concepts. What I've seen in my practice and in some of the talks I've given out there is that, as molecular pathology is becoming more and more common in anatomic pathology, more and more utilized, both at reference labs and at some smaller pathology groups, a lot of people are sort of being thrown into this field who have sort of minimal background in actual molecular biology, both in terms of the MDs that are ordering and interpreting the tests and the various people that are performing them in the lab. So I do think it's quite important for people to sort of get that foundational basic understanding of molecular biology that helps people understand what these things are, as well as the basic foundational understanding of the techniques themselves. So here's a list of some of the common assays that we're starting to see in pathology. You've probably seen some of these being ordered in your various labs. Some of them are really becoming standard of care for diagnostics and prognostication. In lung cancer, I'll just list a few here. In lung cancer, EGFR mutation analysis and ALK1 translocation analysis has become basically standard of care for any stage 4 adenocarcinomas of the lung. In colon cancer, KRAS and BRAF mutation analysis are now becoming common for prediction of EGFR-directed therapy, predicting the ability to use EGFR-directed therapy, and also some syndromic associations, so Lynch syndrome genes are now being tested in many colon cancers. In the past year or so, a particular drug called vemurafenib, or Zelboraf, has now become approved for melanoma treatment, but only in the context of melanomas that are BRAF mutated. So now BRAF mutation analysis has become basically standard of care for any metastatic melanoma. One that has a little bit longer history is HPV analysis in cervical cancer. So, the past decade or so, it's become very commonplace to test for HPV in pre-cancerous lesions of the cervix, and that's a molecular assay in most cases. HemePath is one of the areas of pathology that, that had molecular assays earlier than many others because there are so many specific translocations that basically define many of the lymphomas and leukemias that we know about, as well as some specific mutational assays as well, JAK2 in myeloproliferative neoplasms and c-KIT in some AMLs. So these are many cancer-related applications of molecular pathology. We can also use molecular pathology in the context of infectious diseases, and I'll describe some examples there in the talk. We can use various molecular assays to detect bacterial, viral, and fungal organisms in specimens, and we can also use molecular assays to detect various genetic or inherited diseases. So this is just a short and partial list, but just to sort of show you how many different things are out there and becoming really quite commonplace, which are important to just be aware of. So those are specific assays, and then this is a list of various techniques that can be used in molecular pathology. I won't go over all of these, but just to show you that there are numerous molecular biology techniques, and some of them are worth knowing because they're the most common ones used in pathology. So the ones that we'll talk about today are above the blue line here. PCR, you've probably all heard about. Polymerase chain reaction has kind of revolutionized molecular biology and molecular pathology. We'll talk about several variations of that today, including your standard PCR as well as real-time PCR, reverse transcription PCR, and allele-specific PCR, which is used for mutation detection. DNA sequencing is another common technique in molecular pathology, and others like in situ hybridization, as well as an older method of cytogenetics. These other ones are also useful, but not quite as commonplace, and we won't describe them, but it's worth having heard about them. Comparative genomic hybridization, melting curve analysis. These are older techniques of southern, northern, and western blotting. RFLP, or restriction fragment length polymorphism analysis, loss of heterozygosity analysis, and microarrays. These are all very useful, but a little too detailed for today's discussion. So we'll talk about the top 4 today. But before we talk about that, the part one of the talk will be sort of foundational concepts of molecular biology. And that really starts with the central dogma of molecular biology, which was first proposed back in the 1950s. And the central dogma describes the information flow in a cell. So you've probably seen this before. DNA yields RNA yields protein. This really describes how the information that's contained in a cell is translated into utility, and I'll describe this in some detail here. This is a schematic of a cell. So nucleus here, cytoplasmic membrane here, and cytosol in here. The DNA, as you probably all know, is contained in the nucleus. I like to analogize the data flow in a cell to a computer to some extent. So the DNA you can think of as the hard drive of the cell. This contains all of the information required to make a human, and every single cell in your body contains the entire complement of your entire genome of DNA, all contained in the nucleus. So that's a very permanent storage method, much like a hard drive. But the DNA is locked into the nucleus. So the information from DNA can't really be used to do anything useful until that message can get out of the nucleus. So mRNA, or messenger RNA, is what's used to transfer that information from the nucleus to the cytosol, where really the functions of the cell are taking place. So whereas the DNA can be analogized to a hard drive, RNA is more like a random access memory, or RAM, of the cell, where that data is transcribed onto a different molecule, but it's basically a copy of the same data that's just more easily accessible. So the RNA is first transcribed from DNA in the nucleus, and then those those RNA molecules then move out of the nucleus into the cytoplasm, where they can be used to be translated into protein. And protein is really the workhorse of the cell. Proteins are what do pretty much all of the cellular functions that you can think about. They provide the structure for all cells, by and large. They carry out all the enzymatic functions of the cell, all the biochemical reactions of the cell. And they're the molecules that do all the signaling between cells and within cells. So when you think about all the various functions that cells carry out, it's really the proteins that are doing them, but the DNA that are telling the proteins how to form themselves and how to do themselves. So the DNA is the information source, and the protein is the workhorse, with the RNA being that transfer molecule that tells the proteins how to form. Also, it's important to remember that DNA is a much more permanent information source. So the DNA molecules in any cell are basically there for good. The RNA molecules are a much more temporary information source. Any given RNA molecule is very short-lived. It's created, it moves into the cytoplasm, it's used to make some proteins, and then it's degraded quickly, relatively quickly. So this is just a temporary information source. In molecular pathology, we have various techniques we can use to analyze these different molecules. PCR can be used to analyze both of the nucleic acids, which are the DNA and the RNA. In situ hybridization can also be used to analyze both of the nucleic acids. Cytogenetics, which we'll describe in a moment, can only be used to analyze DNA. And the older techniques, which we won't really talk about today, of southern blot and northern blot analysis, can also be used to analyze these nucleic acids. Immunohistochemistry, which is obviously a very common technique in pathology, is what we use to analyze proteins. In the larger research world, there are other methods as well, like Western blotting. And you could really argue that immunohistochemistry is a molecular technique. We often don't lump it into molecular pathology because it's been around for so long, but we're really using it to analyze molecules in the cell. So it really is a molecular pathology technique. DNA, as I just mentioned, DNA and RNA are both nucleic acids. Their structure is quite similar in terms of what they're composed of. RNA is a ribonucleic acid, and DNA is basically the same thing with a mild or a slight chemical alteration that removes one hydroxyl group, but it basically looks the same as RNA. RNA and DNA are both made up of 4 different bases or nucleotides: adenine, thymine, cytosine, and guanine. So when you see a sequence of DNA, you'll see repetitive A's, T's, G's, and C's. RNA is the same, except that the T's are replaced with U's, which stands for uracil. But basically think of these as the same structure. Proteins are quite different. They're made up of amino acids, And there are 20 common amino acids in humans and pretty much all other organisms, which are listed here. We don't have to go through those in details. They're usually abbreviated with either these 3-letter abbreviations or single-letter abbreviations. DNA is structured as a double helix, and I'm sure you've all seen many times the classic double helix that was first described in the 1950s. One chain of the double helix is a single molecule, which is many, many, many base pairs long. And you can see those schematized over here as T's, G's, A's, and C's. And on one molecule, one strand, those T's, C's, A's, and G's are complemented by a complementary base pair on the other strand, which fits like a lock and key. So for every T, there's an A on the opposite side that fits into the T, and these are by non-covalent associations. And for every C, there's a G on the other side that fits by non-covalent associations. So if you know the sequence of one, you know the sequence of the other is just the complement to it. RNA, like I said, is also made up of nucleic acids, but there's a little bit more variety in RNA. We can think of RNA as two major categories: the coding RNA, which is the messenger RNA, and various forms of non-coding RNA, which does not code for protein, but does various other functions in the cell. We don't have to describe all of these in detail, but I'll just go over them briefly. Unlike DNA, messenger RNA is a single strand. There's a coding region within a messenger RNA and various other portions that do other functions that do not code for protein. Messenger RNA is the one form of RNA that codes for protein. There are numerous other kinds of non-coding RNA that we have listed here. Ribosomal RNA here is the type of RNA that's in ribosomes, and ribosomes are the structures in a cell that do the translation in the cytoplasm. Translation, again, going back to the other slide, being that transfer of information from RNA into forming a protein. Ribosomal RNA is involved in that step. There we go. Transfer RNA is also involved in that step. We won't go over those in too much detail. And microRNA is a more recently described form of RNA. It was discovered about a decade ago. And this is becoming actually more relevant in clinical pathology. MicroRNA is, there's about 500 or so that are known in humans, and they are very short segments of RNA, as the name would imply. They're microRNAs. And all of them form these hairpin structures where in the middle of the structure it flips back on itself, and one side of the molecule is actually complementary to the other side. These are involved mostly in regulation of gene expression. As I mentioned, proteins are quite different from DNA and RNA. The variety of proteins is much, much larger as far as the structure goes, and that makes sense because proteins are carrying out all of the various functions in the cell. These are just a couple examples, but depending on what proteins are doing, they have very different structures. So these are intramembrane proteins. I think these were described in bacteria here. These are ones that are ion channels through a membrane. Various other structural and signaling and enzymatic functions that proteins undertake, and as expected, they show very different structures when you look at them in these schematics. Just again to review the flow of information, DNA yields an RNA molecule, so a gene of DNA will give rise to an RNA molecule that's basically exactly the same as the DNA, except that the T's have been replaced with U's. So there's really no translation of that information. It's just a transcription from one to the next. The RNA is then taken into the cytoplasm where it's translated into amino acids. There's a long list of amino acids, and this translation uses the genetic code, which we won't go into in too much detail, but basically the genetic code takes 3 bases. That's a code for a single amino acid. So every 3 bases codes for 1 amino acid. And then this string of amino acids folds by various interactions from one amino acid to another one, folds into these various structures that carry out the various functions in the cell. So this slide is intended to sort of show the variety of different cell functions that you have in the body, despite the fact that every single cell in the body has the exact same genome. So just a few examples here of different types of cells. These are keratinocytes that make up the skin surface. They function as a barrier to the outside world. This is a megakaryocyte that you would find in the bone marrow that's producing platelets that are involved in coagulation in the blood. And these are heart muscle cells. just look at how different these different cells look and how incredibly different their functions are, making platelets, barrier to the outside world, and making your heart squeeze. All of these cells have the exact same genome, and yet they look completely different, and they perform completely different functions. The human genome, just a very brief review of it, is composed of something close to 3 billion base pairs, or 3 billion nucleotides, which are organized into 23 chromosomes. Of those 3 billion base pairs, only about 1.5% actually are genes that encode proteins. And if you look at the genes in a human compared to our closest relative, chimpanzee, or a more distant relative, the mouse, those genes, and that 1.5% is about 25 to 30,000 genes, those are actually very, very similar among various related organisms. So not only do different cells that have the same genome look and function very differently, but different organisms that have the exact same gene, or not the same genome, but very similar genomes look very, very different. So how can that be? We go back to these three pictures again: keratinocytes, megakaryocytes, and heart muscle cells. Why are they so different? Well, it's all based on gene expression. So as I mentioned, all your cells have the same genes, but they look and function differently, and that's because only a small subset of genes are expressed in each cell type. So for example, when pathologists use immunohistochemistry to try to determine what cell type they're looking at, for example, in a tumor, why that works is because different tissue types only express certain genes and therefore only have certain types of proteins in them. So if we were looking at a sample of skin and we stained for a cytokeratin, it would be positive because cytokeratin genes are expressed in keratinocytes. If we did the same staining on a megakaryocyte, it would not be positive because that gene is not expressed in a megakaryocyte, and the same goes for skeletal or heart muscle cells. So gene expression is obviously very critical in determining what cells do, how they function, and really what organisms we are. So not only is gene expression critical for determining what cells do, but it's also critical in maintaining their normal function rather than becoming tumors. So here are two examples. A normal melanocyte here in the skin doing its normal function of producing melanin and protecting the skin cells from ultraviolet damage. This is a single melanocyte. It looks normal. It's functioning normal. But if gene expression goes awry, it can become a melanoma and be malignant and kill people. These are normal keratinocytes doing their normal protective barrier function, but if gene expression goes awry in them, they can become a squamous cell carcinoma. And this process is called tumorigenesis. Basically, tumorigenesis, think of that as just gene expression going awry. So, and that can happen for multiple different reasons. You can have deletions of genes, so genes that were supposed to be there keeping cells in check can be deleted, so now the cells proliferate abnormally. You can have amplifications of various genes that could cause cells to proliferate abnormally or go into places that they're not supposed to be. In some cases, you have genes that are mutated at specific spots or genes that have been translocated from one chromosome to another and therefore functioning abnormally. Or in some cases, chromosomes that have been duplicated or deleted that causes tumorigenesis. So all of these basically end up resulting in abnormal gene expression, which can result in tumor formation. Most of these changes happen at the level of DNA. You can think of gene expression as happening at that level of DNA going to RNA, which is transcription, but really most of the changes that we're really analyzing in pathology are within the DNA itself. All these deletions, amplifications, mutations, translocations, and numerical chromosomal abnormalities are with DNA. So that's sort of the part one basics, fundamental understanding of molecular biology, and I'd be very happy to take any questions at the end on more detail about that. But I'd like to move on to part two, which is describing some of the common molecular techniques that we use to analyze those alterations in DNA or in some cases in RNA in pathology. So as I mentioned before, there are many, many different techniques in molecular biology. Some of them are now quite important in clinical pathology, and we'll describe these for here today. PCR, again, polymerase chain reaction and its variants, sequencing, in situ hybridization, which has various forms that we'll describe here, cytogenetics. We'll go in reverse order to start, because cytogenetics is sort of the first technique that we can think of as a molecular biology technique that's been around actually for quite a while. Been around even before we had things like PCR. So cytogenetics, even though it's an old technique, is still very, very useful. And we still do it very frequently in, for example, hematopathology. Cytogenetics is mostly used to detect large-scale chromosomal abnormalities. So those would include numerical chromosomal abnormalities, for example, increased numbers of certain chromosomes or loss of certain chromosomes. It can also be used to detect partial large-scale chromosomal abnormalities. So if you lose a portion, you still have a chromosome, but you've lost a portion of it, that would be a partial deletion, or large amplifications that can be detected. And also translocations, where one portion of a chromosome has been moved over to connect to a different chromosome, and you have abnormality at that junction point that can cause abnormal gene expression. So I'll describe these. A couple of important things to know about cytogenetics are that it can only be performed on living cells and on mitotic cells. So this schematic here shows the cell cycle, which you've probably seen before. Most cells during most of their lives are in interphase, meaning the phase of the cell cycle where they're just looking like their normal cell and functioning as they normally would. During interphase, the chromosomes are not organized into the classic X shapes that you're used to seeing, but are actually distributed diffusely throughout the nucleus. So even though you're used to seeing chromosomes as these types of structures over here, really during most of their lives, chromosomes are diffusely spread out throughout the nucleus and can't be recognized individually. So most cells, most of their lives are in interphase, but you can't use cells at that phase to do cytogenetics. Cytogenetics can only be done on mitotic cells when the chromosomes have condensed into these X shapes and can be recognized individually. And because you can only do cytogenetics on mitotic cells, you can only use living tissue to do cytogenetics, because you have to induce proliferation and stop the cells at that mitotic phase to get the chromosomes from them and analyze. And that can only be done if the cells are actually living. And that's an important thing to remember. So when that's done and the chromosomes are analyzed in these cells, you can't see them unstained. You have to stain them with a chemical called Giemsa, and you see that here. So for reasons that are kind of unknown, human chromosomes stain in very characteristic patterns when you put the Giemsa chemical on them. And there are certain regions that are dark and certain regions that are light. And because these regions are very consistent in all your chromosomes, we can actually use that staining pattern to recognize which chromosome is which. So this is called G banding, or Giemsa banding. And we don't have to go over them in detail, but basically it's very well known what the characteristic pattern is, kind of like a barcode of each chromosome. So once the Giemsa is put on there and the chromosomes are separated and analyzed, we can tell exactly which chromosome is which, how many of each one there is, and whether there are any major abnormalities in their structure. And here are some of the abnormalities that we could detect when doing this. So numerical abnormalities would just be, as I mentioned before, an increase or decrease of an entire chromosome. This is an example of trisomy 21, or Down syndrome. So there's 3 chromosome 21s instead of 2. They're not abnormal looking, there's just an extra one. So that's a numerical abnormality that can be detected by cytogenetics. A partial deletion would be removal of a portion of one chromosome. So here you can see this chromosome 5 normally would be this length. Oops, let me go back there, sorry. Normally chromosome 5 looks like this, but in this deletion of 5q, or deletion of part of the long arm of chromosome 5, this is involved in myelodysplastic syndrome, and you can see that as the absence of this end here. A translocation is not an increase or decrease in the amount of any one chromosome, but a movement of one portion to another chromosome. So here, this portion of chromosome 22 has moved onto chromosome 9. So this is a 922 translocation, which is known as the Philadelphia chromosome, and that's what causes chronic myelogenous leukemia, or CML. So these are all large structural abnormalities in chromosomes that can be detected by cytogenetics. In contrast, in situ hybridization, the next technique that we'll talk about, can detect much smaller abnormalities in chromosomes. So the key features of in situ hybridization are that we need a probe to detect a very specific sequence in DNA, or in some cases in RNA. So it's not looking at the entire genome or the entire complement of chromosomes. We're looking at one very specific area. We're visualizing in situ hybridization in situ, as the name implies. In other words, within the context of the tissue itself. We're not extracting the chromosomes from the tissue. We're looking at them actually in the tissue while it sits in place. And critically, ISH can be performed on interphase cells. So we don't need living, viable cells to perform ISH. We can do it on interphase cells, so we don't have to induce them to undergo mitosis. To understand the technique of ISH, I think it's easy to compare it to immunohistochemistry because IHC is a little bit more well known in pathology and it's been around for much longer. And the technique is actually quite similar. So in immunohistochemistry, we have three major elements. A target, which would be a protein in immunohistochemistry, a probe, which would be an antibody, and a detection method, which is an enzyme that yields some sort of colorimetric product. Usually in IHC it's peroxidase or phosphatase. Compare those to ISH, really have the same three elements. You have a target, which in ISH would be DNA or RNA. You have a probe, which is not an antibody, but now it's actually the complementary portion of DNA or RNA that's binding by the known T to A and C to G interactions. And you have a detection method. So in IHC, the detection method, as I just mentioned, is usually using an enzyme to create some sort of color. And in ISH, you can use various other techniques. And here, this one that's shown first is the oldest technique, which would be a radiolabeling. The more commonly used technique now would be a fluorescent labeling. So that when it's fluorescent ISH, we call it FISH. And an even more recently available technique would be a colorimetric assay, or KISH, colorimetric ISH, and that's basically just like IHC, where we use an enzyme to create a colored product. And here's some examples of what they look like. So if it's a radio-labeled ISH technique, then you put a radiographic film on your experiment, and you can see where the radio label has bound. If it's fluorescently labeled, you have to use a fluorescent microscope to detect it and a fluorescent labeling of the background nuclei. And you can use various colors of fluorescent labels like green and red. And if it's chromogenic, we can use more typical histology techniques for labeling nuclei and cells. And we have still various colors of colorogenic products that can be used to detect various probes like blue, red, and brown colored products. So how can we use ISH in pathology? Well, some of the uses are actually quite similar to cytogenetics, and some are a little different. So similar to cytogenetics, we can use ISH to enumerate chromosomes or detect whether there's an increased number of certain chromosomes or a decreased number of certain chromosomes. We can also use it to detect abnormalities of the exact loci or genes in chromosomes. In other words, if there's amplification of certain genes, such as HER2, which is commonly done in breast cancer. And we can also use ISH to detect translocations, just like we do in cytogenetics. Some techniques that we can use ISH for that can't be done with cytogenetics would include probing specific RNA molecules, so specific transcripts like mRNA molecules, or actually specific foreign molecules such as viral or bacterial RNA. And in the future, we may be able to use microRNA ISH. That's actually been described, although it hasn't come into clinical utility yet. So I have some pictures of examples here. So this is chromosome enumeration. A probe for centromere chromosome 21 shows 3 instead of 2, so that's a Down syndrome case. HER2 amplification. So this is a FISH, and we have a red probe for the HER2 gene and a green probe for the centromere of 17 of chromosome 17 gene, or actually just the centromere of 17. So we're comparing the HER2 copy number to the total centromere 17 copy numbers, and we can see that the HER2 has been amplified in a breast cancer case. A translocation analysis where this is a mantle cell lymphoma case where a probe for the locus on chromosome 11 that gets translocated to chromosome 14 in mantle cell lymphoma normally would be separated on chromosomes, so the two chromosomes that are not fused here are separated as expected, whereas the ones that are fused due to this translocation are right together. So you see overlapping of the red and green signals, and that shows you that there's a translocation in this case. So again, those are techniques that are somewhat similar to cytogenetics in terms of what they determine. And then other ones that look for RNA are not similar to cytogenetics. Here we're looking at the kappa and lambda light chains in plasma cells to determine clonality, and this looks like a mixed population of plasma cells. We're looking for foreign RNA molecules. This is a RNA in situ hybridization for the EBV, or Epstein-Barr virus RNA, and we can see that some of these cells have been infected with EBV. This is a picture from a paper that has described a newer technique for microRNA ISH, and again, that's not in clinical utility yet, but we may see that at some point in the future. So to compare the uses and advantages of cytogenetics versus ISH, again, cytogenetics does not have a specific probe, it's just a broad survey of all the chromosomes. So we don't need to know exactly what we're looking for when we use cytogenetics. We can just find any unexpected abnormalities that there might be. ISH, the advantages are that you can do it on interphase cells. You don't need viable or mitotic cells to do it. And we can look at it within the context of the tissue. So it helps us know which cells we're actually looking at, whether we're looking at tumor cells or non-tumor cells. ISH can also detect smaller abnormalities than can be typically detected in cytogenetics. Usually in cytogenetics you need pretty large-scale abnormalities of the chromosome to see them. And again, ISH can detect RNA molecules, which can't be done with cytogenetics. Moving on to polymerase chain reaction, or PCR. Similar to ISH, one of the key features of PCR is specificity, and that's because both ISH and PCR use DNA probes, or DNA primers, which are very specific for your target. So they both have very high specificity. The one feature of PCR that's different from ISH and critical for its utility is amplification. So ISH does not amplify its target at all. It's just looking at one specific spot in a cell in the nucleus, whereas PCR extracts DNA from the nucleus and amplifies it. So it's a much more sensitive technique in most cases. I'll just sort of describe in brief what some of the components of the PCR reaction are and how it works. We won't have too much time to go into real detail of it, but just so you understand the basics of it. There are four main components of a PCR reaction. Obviously, you need a target or template DNA. We also need DNA primers. These are the source of the specificity. You need the raw material for building DNA, which are the nucleotides, the A's, T's, G's, and C's. And you need an enzyme called DNA polymerase, which is what builds a DNA molecule. Those are the four key components, and there are three key steps of a PCR reaction. These are denaturation, which is the melting or breaking apart of a DNA double helix by heating. Hybridization, where the DNA primer hybridizes or anneals to its target. And polymerization, where the DNA polymerase extends off of that primer to form a new DNA molecule. We'll show some schematics of this. So in steps 1 and 2, we have heating and cooling. Heating breaks apart the two DNA molecules into separate strands. Cooling allows them to find each other and come back together. When we cool them, in addition to its native molecule that the two strands could find, we've added these primers to the reaction. So since these primers are in great excess over the initial template, after melting the two strands apart, it's more likely that a primer will bind to its target than the target binding to its complementary other strand itself. So after we've annealed a primer to the target by cooling it down to a point where it can bind, then we raise the temperature of the reaction somewhat to the point where DNA polymerase, the enzyme, is most active. And then DNA polymerase can add the various raw materials, the A's, T's, G's, and C's, on the end of the primer to form a new DNA molecule. So to look at those steps again, we start with the template, which is double-stranded. We denature it at high temperature, where the two strands fall apart. And then the various primers are in the mixture. we decrease the reaction mixture to a lower temperature at which the primers are able to bind to their targets. And then we raise the temperature to a point that's not high enough to make the primers fall off of their targets or melt, but at which the DNA polymerase is now active and can fill in the DNA from the end of the primer. At the end of one round of PCR, then, we'll start from one, let's say we start from one piece of DNA target, and at the end of one cycle of PCR now we'll have two. So we've doubled the amount of that target. In a PCR reaction, we then repeat this three-step cycle many, many times, usually 25 to 35 times, and in each individual step, the amount of product doubles. So that's the source of the amplification of PCR. So again, the key features are the specificity, which is from the primers, which are designed to bind specific areas of DNA, and amplification. And the source of amplification is this repetitive doubling in every round of PCR. We can quickly explain some of the variants that you'll hear about of PCR. Real-time PCR is very similar to what we just talked about, except that it's quantitative. And I'll show you why that is. So if we look at, here we go, a graph of PCR product, because this is a logarithmic graph, so a linear appearance here is actually an exponential increase in the amount of product. So at the end of a long PCR reaction, we have much, much more product than we started with. In your standard original PCR, we would only analyze the amount of this product at the very end, which is easy to analyze because we have a lot of the product. But at the end of this reaction, we've actually lost the quantitative information that tells us how much of the original starting material we had. In these earlier phases of the PCR reaction, these are still quantitatively related to how much starting material we had. So in real-time PCR, instead of waiting to analyze the product until the very end, we analyze the product during the reaction. And if I could just go back here, we do these reactions in these thin capillary tubes where fluorescently labeled various forms of detection devices to detect these products can be used actually at the end of each cycle of PCR. We can quantitate how much has been produced, and that allows this real-time quantitation to show us how much original starting material of the target we had. So really, real-time PCR is the same as regular PCR, except that we're analyzing it during the reaction instead of at the end of the reaction. And then reverse transcription PCR, which is also RT-PCR, not to be confused with real-time. It's reverse transcription, not real-time. Reverse transcription PCR is, again, the same as regular PCR, except that there's one step in the beginning. And the step in the beginning is to convert an RNA target to DNA. So with regular PCR, we're starting with a DNA target. With reverse transcription PCR, we're starting with an RNA target. And that requires the enzyme called reverse transcriptase. And if you think back to the central dogma, the normal flow of information is from DNA to RNA. Normal transcription is from DNA to RNA. So reverse transcription is from RNA to DNA. And this is a special enzyme that we get from viruses that do this, that can take an RNA target and create a DNA molecule from it. So reverse transcription PCR is just a first step of RNA target plus a specific primer plus this reverse transcription enzyme to create a DNA copy of that RNA, and then regular PCR like we just described. So how can we use these various PCR techniques in pathology? Well, in the case of RT-PCR, or reverse transcription PCR, where we can look for RNA, we can use that to detect gene expression in cells, similar to immunohistochemistry, which looks for the protein products of those gene expressions. With RT-PCR, we can actually look for the mRNA products of gene expression. So that can help us determine what tumor types we have, like the biotheranostic cancer type ID test that's used to determine where a tissue came from, if it's an unknown primary in a metastasis. We can also use it to prognosticate. For example, in the Oncotype DX test that's used in breast cancer, that's a reverse transcription PCR test that looks at gene expression and uses that for prognostic information. And we can also use that for detecting abnormal mRNA transcripts. For example, the BCR-ABL, which is the product of that Philadelphia chromosome I described a while ago in CML, that's an abnormal transcript that wouldn't be present in any normal cell. So if a reverse transcription PCR reaction is used to detect the presence of that, that can be used to diagnose that translocation without actually looking at the chromosomes themselves. We can also use either reverse transcription PCR or regular PCR to detect DNA and RNA molecules that shouldn't be there, for example, viral, bacterial, and fungal molecules. And we can use it to detect various mutations in tumors, which I'll describe in a moment. I'll skip these. These are various other uses of PCR that we won't go into detail of. So using PCR for detection of mutation, this has become a more common technique in recent years. For example, some of those assays I described in the beginning of the talk, like EGFR and BRAF. So allele-specific PCR is one of the ways we can use PCR to detect mutations. And basically the concept for this is quite simple. The concept is that in a primer, so this is a primer here. This is a schematic of a PCR primer, this green thing. And of course this would actually be multiple bases here. C's, T's, A's, and G's, about 18 to 25 long. And the important thing to know here is that usually these are designed to be exactly perfect matches to your target. But if you had a mismatch somewhere in the middle here, the primer would still usually bind just fine. And you probably still have a PCR product as a result of it. But the very last base in the primer is actually quite critical for the PCR reaction. And if that base is a mismatch, and doesn't bind correctly, you usually won't get a PCR product. So that fact can be employed to detect specific known mutations in DNA. So in this particular example, this primer has been designed to bind perfectly if this DNA is a known mutated form, where this is a G mutant, and the primer has a C there to bind correctly. Whereas if the wild type or A base is there, the primer won't bind correctly and therefore won't be able to yield a PCR product. And then that can be used in a real-time or any other sort of PCR detection method where the one that bound and created a product can be detected, and the one that didn't and didn't create a product won't be detected. So you know then whether your starting material had the mutant form or had the wild type form. So that's one way of finding mutations. It's a more common way these days. And the older way is DNA sequencing, which is still used in some cases. I'm not going to go into too much detail here, but it's worth being aware of various types of DNA sequencing. The most common one used in clinical applications these days is chain termination sequencing, or Sanger sequencing. Another one that's becoming somewhat common is pyrosequencing. That's also known as sequencing by synthesis, and I won't go into much detail about that today. And then you're going to start hearing a lot about next-generation sequencing, and I'll have a few words about that at the end of the talk. I'd love to go into real detail about how sequencing works, but every time I try to do this, I realize that it would take an hour to really describe this in a way that people really understand. So I'm only going to talk about a few key points about sequencing, and we're only going to focus on that one form, which was the Sanger sequencing, or chain termination sequencing. So I'd just like to point out a couple things about this, and I think the way you can think about this method is basically it's a lot like a PCR reaction. You have three major steps: denaturation, annealing, and extension. The main difference is we only do one round. We don't do multiple rounds that amplifies the target. We do one round to just create one product. And instead of having two different primers that bind at different sites, in a sequence reaction we only have a single primer. So I won't go into too much detail about how this really works, but just know that it basically has the same components as a PCR reaction, with one special extra element, which is a modified form of these nucleotides, which I'll show here. This modified form is a chain terminator, which is it's a fluorescently labeled A, T, G, or C, which has been modified so that it stops the reaction. And just see the schematic here. By stopping the reaction, you end up with a reaction product of multiple different chains at different lengths that have been stopped or terminated at different sites, depending on where that terminator has been incorporated. I know this is a bit complicated, sort of hard to imagine, but basically it's just like a PCR reaction, except that instead of all the same length product, you get multiple different length products because you have these chain terminators in the reaction. And those terminators are fluorescently labeled so that a detector can detect where in the chain these specific terminators have been incorporated. So at the end of one of those reactions, you would run that product out by electrophoresis and detecting the various places that these terminators have been incorporated. And by looking at that, you can see where a C or a T or an A or a G has been incorporated into that reaction. And that tells you the exact sequence of that product. If you're using this to detect a mutation, then we can detect that mutation by seeing aberrant or unexpected peaks in that reaction product. And that's a quick way of looking at it, but I'd be happy to answer any questions about that if people want to talk about the details. The important things to know are that unlike PCR, sequencing does not amplify the product. You're only running it one time. It's not multiple repetitive rounds of doubling. So because of that, in comparison to allele-specific PCR, sequencing is lower sensitivity. So that's a major disadvantage of sequencing for mutation detection. Allele-specific PCR, in contrast, is very high sensitivity because there's massive amplification as a part of it. The advantages, however, of sequencing are that you don't need to know what you're looking for ahead of time. You can analyze relatively long stretches of DNA, and it can detect unexpected or unknown mutations, whereas with allele-specific PCR, you need to know exactly what mutation you're looking for and design those primers for that exact site. And so you need to specifically target a known mutation. And if there are multiple potential mutations, you might need to run multiple different PCR reactions. And if there are unknown mutations that might occur, you might not be able to detect those in an allele-specific PCR. So the major advantages, again, are much higher sensitivity for PCR-based mutation detection, but the ability to detect unknown or unexpected mutations in a sequencing-type mutation reaction. And I'll very briefly just mention next-generation sequencing. Next-generation sequencing is sort of a general, generic term used for multiple new methods of sequencing that are coming out that give us much faster and much cheaper sequencing of large regions of DNA. Numerous different techniques are commercially available or coming out. And I won't describe these methods in detail, but most of them use what's called massively parallel sequencing, so multiple reactions in the same system that are sequencing various stretches of DNA that can then be compared and aligned to each other to give you long stretches of known sequence. And a method of sequencing by synthesis, which is somewhat analogous to that pyrosequencing that I alluded to briefly before. So again, I won't go into these in detail, but just know that there are multiple new methods of sequencing beyond that Sanger sequencing that I just described, which are able to give us much larger stretches of DNA sequencing than we could before. And to sort of explain how effective they are, I'll just mention briefly that the Human Genome Project, which was done about a decade ago, which was the project that showed us what the entire human genome is, that took 13 years and cost $3 billion. That was done by Sanger sequencing. In contrast, these new next-generation methods that are coming out can sequence entire genomes within a day or so for the cost of around $1,000. We're getting down to the $1,000 mark, a few $1,000s for an entire genome, as compared to $3 billion that took 13 years. We can now sequence entire genomes in a few days for around $1,000. So these are really revolutionizing sequencing. It's also worth knowing that although they've revolutionized the ability to do sequencing, the actual utility of that is somewhat in question at this point. So lots of people would like to know their DNA sequence of their genome, but whether we can actually translate that into knowing any sort of future prediction of what diseases you might have, we're not quite there yet. But as we sequence more and more people's genomes and more research comes out on that, and we'll know more and more about what genetic alterations can lead to what diseases, we're going to start to see in the next decade or so much more clinical utility of whole genome sequencing with these next generation methods. And I will stop there. Hopefully most of that was comprehensible, and I'd be very willing to take any questions that might have arisen.
Thank you so much. That was a well thought out and well presented presentation full of a lot of useful information. I really thank you for taking the time to put together such a great presentation. At this time, I would like the audience to type any questions they have into the question box, and we would go ahead and address those. But as we're waiting for the questions to come in, I would just like to remind you all of the upcoming webinar. And as with all of our webinar presentations, we would like to share a featured product with you. And this one is rather exciting for us because it's a brand new product that is hitting the market now. It's a fully automated smart microtome. It features auto-align and auto-trim technologies. It's one-touch trimming and programmable sectioning. It standardizes section thickness to help optimize IHC and ISH or ISH. And it's the most accurate and precise microtome on the market today. It's one step up, or it's the next generation of the automated microtomes that have been previously on the market. Your Sakura reps do have information on this, so if you're interested or you would like to see what it's all about, please reach out to them directly. Well, thank you for your time, and I'd like to thank the audience for attending. We did have a lot of labs log in today, so I just want to thank everybody for their support and the webinar series.
Okay, thank you.
All right. Thank you. You have a great day.